In the grain belt, there's a vital, thriving community that we know very little about. Where is it? Well, it's right beneath our feet in the soil. But a new soil quality monitoring project currently being rolled out right across the nation will help grain growers to better understand who makes up this subterranean community and how its members function.
Our grains industry has long recognised the importance of the soil, its physical, chemical and biological makeup. And much research is concentrated on the physical and chemical aspects. Now, with advances in molecular research, work that is unravelling the DNA of our soil microbes, we're starting to unlock the secrets of a new world.
So a handful of soil has about 5 billion microorganisms in it, so that's about the same number of people on Earth. And we can measure that as a mass of organisms, and that's what we call the microbial black box. But it's a single number, it tells you something, but not a lot. And these molecular techniques are now really allowing us to open up that black box and explore the different functionality, the different populations that are really important.
Daniel Murphy is leader of the Soil Quality Monitoring Project within GRDC's Soil Biology Initiative, a project that's being rolled out across the nation to help growers better manage soils from both a sustainability and production point of view by understanding soil's microorganisms, the functions they perform, and how managing the system can influence grain performance.
And if you think about it, although we put fertilisers onto crops, it's actually the microorganisms in soil that are involved in all that organic matter breakdown that are re-releasing all of those nutrients back through to the plants every year.
Doing that microscopic work below the soil surface is 90% of the planet's organisms, a community we know very little about.
Not very much at all, and that is probably a reflection on the techniques that we've had available to us to be able to, say, unravel or pull apart the soil biota. It's always been a bit of a black box, the whole area of soil biology. So now a few things have come together to allow us to pull apart and see what's in the soil. So techniques of involving, say, DNA-based approaches are allowing us to look at the different species that exist in the soils.
Pauline Mealy oversees GRDC's wider soil biology initiative, part of the program led by Martin Blumenthal, Senior Manager, Natural Resources at GRDC. Phase 1 of the initiative set the groundwork for this second phase, on which projects including soil quality monitoring are being built.
Farmers often say to me that it's the last frontier, and we have to agree because, relatively speaking, we don't know much about what's happening in the soil in terms of the interactions between different groups of organisms and how that then impacts on the capacity of the plants to grow well.
So we often think of the microbial population as like the eye of a needle, so it's a very small hole, it's a very small component, but it's through which all nutrients must pass. So they're really the powerhouse of the soil. They're breaking down the organic matter, re-releasing the carbon dioxide back into the atmosphere and completing the carbon cycle. And as a byproduct of that, all of those nutrients, whether it be nitrogen, sulphur, phosphorus, macronutrients, are also being re-released and becoming plant available.
Billions of microorganisms in a handful of soil and billions of handfuls of soil. Just as soils vary across the nation, so too does the biological population of those soils. And the advent of molecular techniques is also allowing researchers to identify and study the function of those diverse microbial communities. Within the Soil Quality Monitoring Project, other sub-projects take the research deeper into the biological community.
I'm probably using technology for soil monitoring that hasn't been used for this type of program before.
At DPI Victoria's Bioscience Research Division, a next-generation Illumina sequencer dives deeper into the black box of the soil's biomass.
The Illumina sequencer here behind me, basically it has 16 compartments that do the sequencing, and each of those compartments we can look at up to 300 million individual sequences from different microbes. These sequences don't just give us information about who's there, they give us information about what these guys are doing. So to explain that a little more simply, that basically if we imagine a plant in the paddock and it's relying on nitrogen fixation, or it's relying on microbes to break down residues and supply nitrogen, so mineralisation within the soil. There's individual genes within soil microbes that are responsible for these functions, and with this type of technology we can actually sequence all those genes.
Just as the Human Genome Program delivered an incredible leap forward in scientific knowledge, Pauline Mealy believes this sequencing of microbe DNA could do the same for soil biology.
Craig Venter from the Human Genome Sequencing Program, he basically did this global ocean survey, and from the DNA that he generated from all the samples, he was able to identify 20 million new proteins. So what does that mean? Well, it could potentially mean that there might be antimicrobial agents that can come from those proteins that can go into the medical industry. There could be genes associated with resistance to high salt concentrations that can be used in whichever ways. In terms of the agronomic applications, just by identifying new genes, say associated with nitrogen fixation or phosphorus solubilization, we could perhaps turn those into applications that will be of considerable advantage.
The soil is an even richer biological environment than the ocean.
Soil's the most diverse habitat on Earth, so there's millions of different species in there, all of which are performing critical soil functions.
Providing far more knowledge about the processes soil organisms carry out is one of the major benefits the Soil Quality Monitoring Project will deliver to grain growers, according to South Australian soil scientist Annie McNeill.
They exude gums and slimes which stick the soil together. They convert organic matter into nutrients which plants can take up. And also that slimes that they organisms might, as they stick these soils together, I guess that it improves their water holding capacity too. So they've got a nutrient advantage, they've got a water holding capacity advantage. So I think that those are the things that perhaps they didn't understand in the past, and but they are beginning to understand because we're able to tell them that there is a range there. There's a range of organisms that can carry out those things. And don't forget as well, they know there are organisms there that are causing diseases in their crops, root diseases, but they're also now aware that there's, from DNA tests, that there are also a lot of other organisms there, the good guys that are battling with the bad guys and actually coming out on top, and they know that they can actually make changes in those organisms from their management practice.
At the South Australian Research and Development Institute, DNA-based new tests for soil organisms are being developed. Tests which can be added to the suite of soil tests ARDI already applies in the Predictor B package used by grain growers and farm advisors.
That's what we hope will occur in the future, so we'll get not only a measure of what the pathogens are in soil, but a measure of some of the beneficials, and that information will be then used to, for consultants to sit with growers and assist them in making good decisions about which way they're heading. And it will also be included as part of the soil quality monitoring program in the long term.
Those beneficials will be free-living nematodes, which occur widely across the nation and are a good indicator as they're very responsive to the changes in nutrient status and soil structure.
Soil is very difficult to monitor, and particularly biological status, and the methodologies we've got for monitoring are often very time-consuming and require a very high degree of expertise in identification of specific organisms. There's very few people in Australia, for example, who can identify free-living nematodes, and these nematodes are the ones that don't cause plant disease. They're ones that exist and are involved with nutrient cycling and with feeding on other organisms in soil. So it's very difficult to understand and to identify things in soil, and so the use of DNA-based technologies, I think, is really powerful in opening up our understanding of what's happening in soil. And in this case, I think we've got some quite targeted groups to look at.
These free-living nematodes are so hard to taxonomically identify.
Catherine Linsell is one of the few people globally who will be involved in the creating of the necessary DNA technology to study nematodes in relation to soil quality.
One of the key things we're trying to do in our project is to try to invest in a young scientist, Catherine Linsell, to develop her skills in this very specialised area of nematode community analysis, as well as get her involved with the development of DNA tests. And we think that's really important because the skills about nematode identification and understanding free-living nematode communities reside in a very few people. And a key collaborator in the project is Graham Sterling from Queensland, and he's working very closely with Catherine to help transfer and develop some of those skills in a younger scientist.
In Western Australia, the Soil Quality Monitoring Project has been running for 5 years. Some 1,500 sites have been benchmarked throughout the state's grain belt, and a website allows growers to get information about their soil and its condition.
The data is presented in a number of ways. So a grower can go in and, if he's involved with the project or even if he has their own values, can plug that value in and compare that value or himself to other growers in the same area, same group or same region to see where he sits in comparison to other people. On the same level, the grower group as a whole or on a regional basis can go in there and look at the data and say, well, what's pH doing in our region? Produce a graph that gives you a pH range and the number of sites in that region that fits in those critical pH range. So yeah, the way we try to present the data in as many useful ways as possible.
The soilquality.org.au website is part of the national rollout and will eventually include soil data from all states. That data will be updated as soil collection teams return to sample sites to see how quality is changing through time and better understand the key management practices that are improving the soil and those which are detrimental.
If we're recognising that there's a management practice that's working, is it feasible to actually implement that at a wide scale, or is it really not feasible, as in the production gains wouldn't be large enough? So we've got to think about it in terms of production. It's about making money at the end of the day. We've got to have a sustainable agri-industry, and to do that we need to make sure we maximise our gains, and part of that is making sure there's no constraints to production in the soil.